Allow me take this opportunity to welcome you back to this other session of ours. Now we are going to look at what we call the Hatch slug cycle or the C4 pathway. So we looked at what we call the Kelvin cycle or the C3 plants. Now we are going to look at majorly the C4 what? The C4 plants. Now we are saying that the carbon 4 plants or the C4 plants are those plants in which the first product of carbon dioxide fixation is a 4-carbon compound called the oxaloacetate. Now the other one for Kelvin cycle or the C3 plant, the other one was the 3-carbon compound. Now this one is a 4-carbon compound which is called the oxaloacetate in bracket OAA inside the mesophyll cells, which is later reduced and exported into the bundle sheath cell for further metabolism. We shall look at this. So now we are looking at the examples of C4 plants, and we are saying examples of these C4 plants. We have maize, sorghum, amaranthus, sugarcane, the Bermuda grass, the Rhodes grass, the nut grass. So we are saying that the C4 plants represent only 3% of the world's vascular plants, yet they contribute about 20% to the global primary productivity because of their high productivity. Now we look at the habitats or where we find these plants. So we are saying that these plants are found in the hot, arid, saline, tropical habitats. So we are going to look at what we call description of the leaf anatomy or the kranzs anatomy, and we are going to compare that of the C3 plants and that of the C4 plants. Now, when they can say what is kranzs leaf anatomy, so we say that is a condition in which the bundle sheath cells and the palisade cells of the mesophyll form two concentric rings or concentric layers around each vascular band of leaves, as you can see down here. When you compare these two, we have what we are calling the one on my left hand side is what I'm calling the leaf anatomy in C3 plants, and that one on the right is the kranzs anatomy in C4 plants. So as you can see there, those two. Yeah, you can pause and draw those two leaf anatomies in the book. Then from there we can go ahead and look at the description of the kranzs anatomy. Description of the kranzs anatomy. Yeah, here we are saying that when we look at description of C4 cycle of C4 cycle in carbon dioxide fixation. So we are going to look at first of all this first step which occurs in the mesophyll. Now we are saying that so this step, the first step, the one we are looking at, we are saying that this step occurs in the presence of the phosphenyl pyruvic acid carboxylase, the one we are calling PEP enzyme. Now we are saying that the phosphenyl pyruvic acid is carboxylated, this one. Yeah, we are talking about this one, that is carboxylated inside the chloroplast of the mesophyll cells to form the oxaloacetic acid. This one is carboxylated. The PEP is carboxylated inside the chloroplast of the mesophyll cells to form the oxaloacetate, the OAA, which is a 4-carbon compound. Now, this 4-carbon compound, we are saying that it is reduced by the NADPH. It is reduced by this to form what we call the maleic acid in the presence of the malate dehydrogenase enzyme. Yeah, then from there we shall see that this maleic acid now enters, or it is translocated or shunted into the bundle sheath from the mesophyll. We are saying that from the chloroplast of the mesophyll cells, the maleic acid is translocated, shunted to chloroplasts of the bundle sheath cells, where it is decarboxylated by NADP to form the pyruvic acid and the carbon dioxide. So the pyruvic acid produced returns the mesophyll cells for phosphorylation by ATP to regenerate PEP, the carbon dioxide acceptor. Yeah. Now we are saying that this maleic here, which is formed, enters, though you can see there that this maleic now enters the what? Enters the pyruvate. So the maleic acid which is formed in the mesophyll now is shunted to the bundle sheath, as you can see here, that this one now enters into, from the mesophyll it enters in the bundle sheath cells. Now when it reaches there, we are saying that now the second carboxylation occurs in the chloroplast. So the first one occurs in the mesophyll cells, as we saw there, and the second one now is occurring in the bundle sheath cells. Now we are saying that now the second carboxylation occurs in the chloroplast of the bundle sheath cells through the Kelvin cycle, and this is what occurs. The rubib of the Kelvin cycle is called the final or the second carbon dioxide acceptor in C4 plants. Then the ribose biphosphate or the ribose bisphosphate accepts carbon dioxide produced in 3 steps and to yield the 3-phosphoglyceric acid as given in Kelvin cycle. Yeah, we are saying that some of the 3-phosphoglyceric acid is utilized in the formation of glucose 6-phosphate and sucrose, while the rest regenerates ribose 1,5 by bisphosphate in the system. So in the Kelvin cycle, 3 ATP molecules are required for the C4 plants, then 5 ATP molecules are required for the fixation of carbon dioxide. So for the formation of glucose molecule, C4 plants require 30 ATP, while C3 plants require only 18 ATPs. The ATPs are adenosine triphosphate. So now in this car system, so we are saying that now this maleic acid which is here, now it goes under what we are calling the second carboxylation occurs now in this to form this rubib. Now the pyruvic acid, which pyruvic acid now moves from the bundle sheath back to what we call the mesophyll cell. So and the process continues like that. So I hope you have understood the description of the kranz anatomy. So we are going to look at what we are calling, or allow me end here, we shall look at the advantages and the disadvantages of C4 plants. I therefore sign out.